Hello, today we're going to be looking at our Unit 1, Lecture 4 over bacterial characteristics. So we've already talked about some general cell characteristics, and now we're going to take a look at those characteristics that apply to bacteria specifically. So starting out, what we want to recognize is that every cell is somewhat defined by a shape. Not every cell can be easily defined, but when it comes to the bacteria, most of them have a relatively straightforward kind of structure as far as the shape and the cell are concerned. So we have names that are used as a basic form of terminology, and this is not only useful just for understanding them, but it's also useful in diagnostics. So in a lab, in a medical setting or technical setting, you can have an analyzation of a culture that comes back to the doctor or the physician in reference to a variety of factors. And shape is usually one of those. So they'll look at the shape, what we'll call the arrangement, and as well as things like flagella and Gram reaction, amongst others. So shape is a common way for diagnostics. So what you want to recognize is that there are three common shapes and then a variety of regular variations of those. This does not include everything, but it does include some of the most common. So coccus means a circle. Rod is a rod-shaped. This will also be referred to as bacillus. So rod and bacillus. And then curved are cells that have some kind of spiral or slightly rounded but not fully circular kind of shape to them. Within the variations, you have these basic shapes, and then within those you have common variations. So Coccobacillus is really just the combination of the rod-shaped and the cocci cells. Vibrio is a more pronounced kind of curved cell. It's really a combination of a rod and a curved shell. Spirillum is a spiral-shaped, a particular variation of a curved cell, and a spirochete is another variation of a curved cell. And these are often referred to as something like a spring-like or even a corkscrew variation that we'll see examples of here in a minute. This is a figure out of your textbook, and here you can visualize cocci, rod-shaped, vibrio, which are a variation of curved cells, spirillum, spirochetes. These are some of the common curved and then variations thereof. Now, in the common list, we don't often include branching, but this figure does include a branching variation that you could include in that list there. Note that not every cocci cell is perfectly spherical, nor is every rod-shaped cell the same kind of general rod shape. So these are general depictions, not necessarily a concrete, accurate description of the shape. Now, there's also a group of bacteria that can be difficult in that they don't conform to a specific shape, and these are what are called pleomorphic, sometimes called polymorphic. Poly and pleo can be used somewhat interchangeably, but the most common term here is pleomorphic. This basically means a variety or more than expected number of shapes. Some bacteria, as they grow, they don't conform to a specific type of shape, and this is what gives rise to the term, and it can make diagnostics a little more tricky given that the shape doesn't adhere to a specific form. The next term that you want to be familiar with is bacterial arrangement. So shape is always in reference to an individual cell. Arrangement, however, is in reference to a grouping pattern. So shape, cell, arrangement, group. These terms often apply along with the shape. So, for example, you might see what's called a diplococci. Diplo is in reference to the arrangement, cocci is in reference to the shape. So diplo means two, cocci, circular cells joined together. Now, some of these terms get a little more confusing because they go back to some of the Latin derivatives. Staphylo is in reference to a cluster. So staphylo means clusters of cocci. Strepto means a chain. Streptococci are chains of cocci. Tetrad is 4. This is a group of 4 cocci. And Sarcina are almost layered tetrads. They usually occur in like a cube-shaped kind of pattern. So I'm not going to make you memorize all of these here, but these are common cocci arrangements. Singular, by the way, is in reference to the fact that all the cells are individual, therefore there is no grouping. So it could be a grouping or a lack of grouping. Rod-shaped cells don't have quite as many arrangements. Here you'll see singular, diplobacilli, streptobacilli, and a unique arrangement here called palisades. Curved cells, as it says, very often do not group together and are almost always singular, and therefore we don't have terms for those. So you have the 3 common shapes and the 3 and the common subtype of arrangements there for each of these different groups, in particular cocci and the rod shapes. Now, of those, this list right here on this figure, these are the ones that I really want you to be familiar with. And what I'll try to do is give you basic diagrams so that we're not trying to get tricky with what we're looking at and just focusing on, do you understand how the pattern and the shape relates to the name, the shape and the arrangement. So for example, if you look at this figure right here, this portion of the figure, which is a cluster of circular cells, if you didn't have the name above it, would you be able to recognize that? If you see a chain of cocci without the name above it, would you be able to recognize that? So be familiar with these terms here and expect to have some kind of matching or identification-based question. Next, what we're going to get into are the features that pertain to the individual cells. This figure in 4.1 is a general reference. It's not meant to look like any bacteria in particular, but is a reference for all the features that could be found on bacteria, of which you can see here there are quite a few. We'll talk about most of these, but this is just a reference to follow back on if you need to pinpoint something or visualize it in the context of a cell. Starting from the outside, moving back here, starting from the outside of the cell, we'll have a variety of appendages, such as one you see here, this big long green noodle called the flagella. Some of these little hair-like structures over here, these are going to be examples of appendages. Kind of like your arms and your legs are an appendage. So an appendage is something that sprouts from the surface of a bacteria, and ultimately they serve one of 2 purposes. They can either allow the bacteria to move with motility or attach. And kind of like your legs help you move and your arms help you grab onto things. Overly simplified, but that's kind of the idea. What we're talking about when it comes to appendages are generic features, and as it says down here, not all appendages are present in all forms of bacteria. So in the case of bacteria, what we're basically describing is what you could see and not necessarily what you'll see on every single species. So one of the most pronounced appendages is the flagella. Maybe you've heard of a flagella, but it's essentially a motility appendage, something that allows the bacteria to move, kind of like a tail for a fish almost. Usually requires some kind of wet environment, and kind of like a fish would whip its tail, the bacteria flagella serves as a similar kind of motility. It looks different, but it functions in a similar way. You can see here some of the context back to shape. All spirillum have flagella, about half of the rod-shaped cells, and only a small number of cocci. Once we have flagella present, the next thing that you can look for is whether or not they occur in a particular arrangement or not. So flagella will come in one of 4 basic arrangements, and that's discussed in some of the textbook questions about bacterial flagella arrangements. So be familiar with some of those from the textbook. Okay, the next part of the bacterial flagellum has to do with the way that it connects to the cell itself. So down inside the cell, let's just go back here real quickly. So if we look right here in this area, this is a flagella, and where it connects to the cell right here is what we're looking at in this figure. So this is that green long tail, And as it comes in contact with the cell itself, it kind of embeds itself down inside the membrane. And from there, we can see that it's segmented into 3 distinct parts. So you have what's called the basal body, and this is what is actually embedded down inside the cell. You have what's called a hook that connects to the last part, which is the filament, which is actually the long tail, the tail-like extension itself. So basal body, hook, and filament. For this class, we're not going to break it down much further, but if you'll note that the basal body itself consists of multiple parts, such as rods and rings. As a real quick visual, the flagella is actually quite interesting. From an animation you can see here in the top, this from a YouTube video, this would be the surface of a cell. This is the filament here, which would extend out, and as it zooms in here, it'll show you what we would expect to see if we were looking at a real bacteria. So here's the hook connecting down into the basal body, and what happens is these rings actually rotate almost like a motor, and they create a sort of torque or tension that is then transferred up through the hook and into the filament, and that causes the filament to wave around and cause the propulsion-like movement. But if you'll note that what you actually have are a series of ring structures that all sort of lock in together and move and rotate, providing motility. It's actually a really fascinating little structure there. For this class, we don't need to focus on every detail, but it helps you visualize what's going on there. So the flagella is your main motility appendage. It's in fact really the primary way. There's always exceptions, but that's really the primary way that bacteria move if they have a flagella. Now, when it comes to attachment, bacteria can attach in a more diverse number of ways, and that starts with a secondary use of the flagella. So the flagella can also serve as a way to attach if the context applies. What that is will be something that comes up in different discussions, but as a general rule, just understand that flagella as its primary function is for movement, but also serves for attachment as a secondary function. These next two down here are really more primarily used for attachment. Fimbriae is really what they do. They are an attachment mechanism pretty much exclusively. And then what's this last one here is called a pilus. This one is used for attachment and genetic transfer, which we'll talk about briefly here in just a minute. So these are all attachment mechanisms that are in the form of an appendage. Fimbriae are considered small bristle-like fibers. So when you hear a description describing it as bristle-like, you'll know that we're referring to Fimbriae. And what happens is that they sprout off from the surface of the cell, and they act as a way to kind of stick to other surfaces in a way that causes the bacteria to attach. This can be done to other bacteria or to the surface or both. And so you can kind of see here, this is the intestinal lining of an animal. You can sort of see the Fimbriae, not real well in this figure, but you can see that this bacteria cell is sticking to the intestinal tract. And then you can see that some of the cells can actually stick on top of the other cells, kind of like you see here. So this is one of the primary ways that bacteria anchor themselves to an environment, and that allows them to really stay in one place rather than being moved around, which is what tends to happen if they can't stick in one place. The pili is a unique appendage. It can serve as just another way to attach, and in that sense, it's another form of attachment. However, the pili can serve a more specialized function under the right circumstances, and what this function is conjugation. So pili can essentially facilitate conjugation. Conjugation is a process in which bacteria transfer DNA back and forth to each other. You can see this in a real image here. This is a real microscope image. You have several bacteria, and you can see here a series of pili connecting to these various cells. Now within that pili is a tubular sort of hollow space, and once it makes the connection from one cell to the next, what can actually happen is the bacteria can transfer genetic information through that pilus into the next cell. And this is a very unique concept. We don't see this in animals or most higher level animals for that matter. So it's a unique process that bacteria go through, but it allows them to quite literally swap their DNA back and forth, absent of some kind of sexual or asexual replication. So you can pass your DNA on to your offspring through replication, but you can also pass your DNA on to the cell that just so happens to be sitting next to you through conjugation. What this allows bacteria to do is adapt and mutate pretty rapidly by sharing information. And that's a concept that will come up later, but for now, we'll understand that conjugation is a mechanism of transferring DNA, and it involves the pilus. That's what we want to know for now. The next thing is the surface coatings. So bacteria not only can attach through appendages, but they can also attach and even protect themselves with surface coatings. So surface coatings will, as it says here, protect the bacteria and aid in attachment. So this is now another way for attachment, but not via appendages, instead via surface coatings. So they provide attachment and protection. The next thing we want to know is that surface coatings occur in two basic forms. The first one, called an S layer, is a thin layer strictly for protection. And this next weird looking word here is called a glycocalyx, or for plural, glycocalyces. So glyco is in reference to sugar, and calyx or calyces for plural is in reference to a matrix. So this actually stands somewhat loosely for a sugar matrix. And so what it is a series of sugar-like substances secreted from the cell that act as protection and attachment mechanisms. Let's talk about the S layer first. The S layer is often described as sort of a chainmail-like structure, kind of like the chainmail armor of a knight. It has a thin but strong layer that simply helps the bacteria with an extra layer of protection. Slime layers and capsules, going back here just real quickly, these are the two examples of the glycocalyx. So a slime layer, as the name accurately implies, is a loose sort of aggregate of the sugary substance, and the capsule is more of a tight, rubbery kind of configuration of that sugary substance. And so you can see a couple different angles, or illustrations rather, shown here, and then a cross section here. So you can kind of see it better in this figure. The idea is that this sugary secretion kind of takes on a series of tunnels and becomes kind of like a matrix-like structure. Backing up here, just and repeat myself one more time, these really help bacteria significantly when it comes to their ability to survive and attach and remain in one place. Okay, the next thing that we're going to get into are characteristics of bacteria that go beyond shape, arrangement, and the appendages or surface coatings. When we peel below the surface of a bacteria, below the appendages and below the surface coatings, what we'll start to see are multiple layers that make up what's called the bacterial cell envelope. Let's just go back here real quickly to the figure in the beginning. If we look at this portion right here, we can kind of see a nice layering pattern. So here we see some appendages. These are fimbriae here. This surface layer here is a coating. This layer right here is an S layer. And now the layers beneath that, here, here, and here, these are getting into the cell envelope. So the cell envelope is going to have potentially an outer membrane, a cell wall, and this last layer here is a cell membrane or a cytoplasmic membrane. So we can have 1, 2, 3 potential layers that you can see here. So these are basically in between all the surface coatings and then the internal parts of the cell. Now, where this is interesting is that from a diagnostic standpoint, The layers can occur in two variations, two of the most common variations. And based upon the person who discovered this, Hans Christian Graham, it was named as either gram-positive or gram-negative. So he decided that that was the naming system. Gram-positives are bacteria that are going to have two layers. One is the membrane, the cell membrane, and the other is a cell wall, shown here in brown. And it's a very thick cell wall. So a thick cell wall, no outer membrane. For context, gram-negatives have the cell membrane, a cell wall shown in brown here, and an outer membrane, which is shown here in tannish plus a purple color. So these two layers are called the outer membrane. This here is the cell wall, and below that is the cell membrane. So depending on what group you're in, the bacteria will have one of these two characteristics. They both have a cell membrane, but whether you have just a thick cell wall or a combination of a thin cell wall plus an outer membrane is what determines whether you're gram-negative or gram-positive. And this is another very common diagnostic characteristic, along with cell shape and arrangement, as well as flagella. So usually minimally you'll want to know what is the shape, what is the arrangement, and what is the gram reaction. And then if you can find flagella, you'll want to know that information as well. So these are all not only just useful to understanding them, but also diagnostics. As we'll talk about a little bit more in later classes, there are implications for how these cell walls and the structure, how they all come together. This is discussed in the textbook review as well. But these structures do have implications for the way that they interact with the body and some of the ways that we treat them with medications and things like that. So that'll come up in the textbook review. The cell envelope is the three potential layers that make up that layering I just showed you a minute ago. The cell membrane is what occurs closest to the inside. The cell wall occurs just outside of that. And for gram-negatives, you'll have an additional layer called the outer membrane, which is exterior to those first two. Note that the cell membrane is found in all life forms, and so that's why we see that in both groups. It is actually common to every kind of life. The cell wall in bacteria is made up of peptidoglycan, and if we go back to that figure here, you can see that they've actually identified this thick layer as peptidoglycan. It could just as easily be called the cell wall, but that's why they're calling that peptidoglycan, because that is the primary building component of the cell wall. And as it says here, whether you're positive or negative determines the thickness. And then the outer membrane is what we see just in the gram-negatives. So the three layers here collectively make up the cell envelope. And so a question that I might ask you is, how does the cell envelope of a gram-positive differ from the cell envelope of a gram-negative? So to answer that question, you would discuss the differences primarily between cell wall thickness and the presence or absence of the outer membrane. So that's how you would go about answering that question. One of the next things we want to talk about with bacteria is a class of bacteria that are referred to as endospores, or endospore-forming bacteria. And with the endospores, what we have is a specific group of bacteria that are capable of producing dormant bodies. And these dormant bodies are extremely medically relevant and relevant to many aspects of human life. And the way it works is most bacteria simply live in one state. We don't normally clarify or classify it, but you might think of this as what's called a vegetative state. Now let's stop for just a second. Let's look at the definition. In this context, the vegetative state means metabolically active and growing, essentially normal function. I just want to point out really quickly that if you think of a human being in a vegetative state, you don't necessarily think of them as being active. You think of them as being inactive. So I just want to point out that this term is somewhat contradictory to what our normal sense of that word is. I didn't make up the definition, but that is the way the word is applied here. So vegetative is actually active. So in a normal group of bacteria outside of the endospore class, they're just always in the vegetative state. However, when it comes down to endospore bacteria, suddenly we now have a differential state. We can be in the vegetative and or the endospore state. And this is where we have a dormant and highly resistant stage. What this means is that they'll be very difficult to kill in the endospore stage. And the way it works is that bacteria will alternate between the two states depending on what's happening in the environment. And it's usually the case that once the bacteria sense there's low nutrient levels, they'll essentially go into a process called sporulation, which causes them to transition from a vegetative to an endospore stage. You can see that here in this figure. This represents the vegetative state. This represents the vegetative cycle. Under normal conditions, the cell will replicate and continue in the active stage. When low nutrient levels are determined, the cell alternates to the sporulation stage. The endospore forms inside of the cell, and that is the only part of the cell that will survive, assuming that the environmental conditions really are detrimental, which is why they typically form these. The active vegetative part of the cell will die, but the endospore state will remain intact. It's kind of like a seed, is a good way to think about it, but way more difficult to kill. So this becomes like a dormant little seed with a tough outer shell on it. And in this state, the rest of the cell has died theoretically. The endospore is now still there, and it can stay in the environment for essentially any period of time. There have been studies where they've found these from ancient fossils that they determined to be hundreds of millions of years old, and the endospore was still active, amazingly. So they seem to be almost indestructible. I don't think they would typically last that long, but given the right conditions, they could theoretically last forever. So hundreds of years is not unreasonable. It just depends on where they land. So the point is that in that stage, they're almost indestructible and they can survive for a very long period of time. So here you can see some figures showing the normal vegetative state, the beginning formation of an endospore, and the later stages of an endospore forming here in an actual microscope image. Once an endospore forms, it'll stay that way for long periods of time, but what will normally happen is it will actually revert back into the vegetative state, as you can see in this figure. This is the endospore here, and this is the vegetative state emerging out of that. And that typically happens when water and a small amount of nutrients are reintroduced back to the cell. So when they lose nutrients, they go into the endospore, and when they get nutrients back, they'll go back into the vegetative state. And so this allows them to basically hide out while nutrients are low and wait it out until the nutrients come back. And by the way, that's very common for bacteria. It's very common for nutrient levels to fluctuate even from day to day, but certainly from month to month or season to season. So it's an adaptation and a survival strategy that allows them to survive harsh conditions. Now, from a significance of humans, endospores are literally the most difficult living organisms to kill on Earth, or the most difficult living structures, the structures of a living thing. They are essentially so difficult to kill that you actually have to take actions to a certain degree just to remove endospores. In other words, if it wasn't for the presence of endospores, microorganisms would be a lot easier to kill. But because endospores are always mixed in with other kinds of microorganisms, in order to truly sterilize something, you have to take it to an entirely different degree just to kill the endospores, because they're so much more difficult than everything else. This is almost always accomplished by high levels of heat for prolonged periods of time. So in order to really sterilize something, you have to put it into a special apparatus that heats it for 20 to 30 minutes at a very high temperature. Most things do not require anywhere near that much and can be desterilized within probably a few minutes at boiling temperatures. But because endospores are so much more difficult, in order to sterilize some kind of whether it's a medical device or even food for canning, as we'll mention here, the actual temperatures have to be higher and more extended in order to compensate for endospores that are very common in the environment. So this applies to surgical procedures. Anytime something is going to be sterilized for an operation, it has to be heated to an excess in order to kill endospores along with everything else. This is also true for food canning. When we can our food, it's shelf stable at room temperature. And in order for that to happen, you have to basically sterilize the food in a way that's actually quite similar to the way that you would sterilize some kind of surgical device. That actually degrades the nutritional value quite a bit. And that's one of the reasons why canned food is not necessarily ideal for getting large amounts of nutrients. Canned food is good for storage, but it doesn't have the most amount of nutrients because by canning it and sterilizing it, you actually degrade the nutrients in that process in order to maintain sterility. So endospores are actually very significant. When we look at these characteristics all together, one of the things I haven't mentioned as much as right now is that these are very common. So endospores are not rare at all. We'll actually do a lab at some point in the semester where we test this. But you can find endospores in almost every location. They're extremely common. So they're not rare at all. And in fact, they should always be assumed to be present because they probably are. So that's the first thing. As we mentioned, they're very difficult to kill. So that's kind of what we've mentioned so far. It requires large amounts of heat. And the last thing is certain species are capable of infection and disease as well as food spoilage. So they're not necessarily benign. Not every species is harmful, but in the endospore class, there are a number of harmful species, and those species are absolutely dangerous and have to be accounted for. So when you put these characteristics together, the fact that they're common, difficult to kill, and potentially deadly, it makes for a very nasty combination of traits. So that finishes up the bacterial characteristics section here. The textbook will go through some of the other information that I haven't hit quite as much, but between this lecture and the textbook review, you should get a pretty good rundown of Chapter 4. All right, thank you.